Alfredo, and today you will hear about the cloning process and the debate surrounding cloning. Since the beginning of memory, human beings have wanted the ability to replicate into exact copies of ourselves. It may be the loneliness that a person feels after losing a close family member. It could be the frantic search for a new organ that a doctor can't find but could save a life. Or it could be the replication of historical figures that would result in students being able to actually talk to a president or historical figure. Clones have been a forethought in our minds for centuries, and as a result, we have created movies such as the aptly named Clone Wars, the Academy Award-winning Jurassic Park, and countless others to bring this dream even closer to reality. Not only is this idea of cloning present in movies, the birth of robotics and artificial intelligence, or AI, all stem from the idea that copying humans is a possibility. With the technology advances of the 20th and 21st centuries, we have become closer to realizing that the dangling carrot that is human multiplication and replication. Now, before the actual speakers begin, here's a brief history of humanity's search to create the biological facsimile. In 1885, a two-cell embryo was split, creating two identical sea oceans. Now, this is the first example ever of artificial cloning. In 1902, another two-cell embryo was split, resulting in a pair of identical salamanders, the first ever artificially identical vertebrates. In 1952, the first successful nuclear transfer occurred, resulting in multiple tadpoles. This is a method that could be used in future with organs. In 1996, Dolly the lamb was born, the first mammalian ever to be cloned. She went on to live a normal life and had 4 children. And in 2013, the human embryonic cells were created from human somatic cells, providing a pathway into actual human cloning. As technology advances, the possibility of society intertwined with human clones is becoming an approaching reality. Now, I would like to introduce the next speaker, Christiana Kyles, who will discuss in depth the cloning process.
Hey y'all, today I'm going to explain the process of gene cloning. When starting the process of gene cloning, you first need to isolate the DNA that contains the desired gene. The DNA is cut into gene-like pieces with restriction enzymes. These enzymes are found in bacteria. Each restriction enzyme recognizes a different and certain DNA sequence. When these enzymes recognize specific sequences of the nucleotide sequence, it breaks the bonds of nucleotides in the DNA strand. When split into two ends, they form cohesive ends. Bacterial plasmids are cut by the same process with the same restriction enzyme. The gene-sized DNA and then the plasmids are combined into one test tube. The cohesive ends of the plasmids and gene-sized DNA are complementary to each other and therefore combined together. This is known as the recombinant DNA. The recombinant plasmids are then transferred into bacteria. The bacteria is grown into colonies on plates and is called the gene library. When the bacteria colony containing the desired gene is located, the bacteria can be multiplied to make millions of copies of the recombinant plasmid that contains the gene. Throughout the knowledge of gene cloning and genetic engineering, people have cloned quite a variety of animals. For example, Dewey the deer, the first white-tailed deer clone, and Dolly the sheep. The process of picking a target gene, isolating it, and then doing the same with the plasmid and connecting the two together and putting it in bacteria to produce the gene helps humans find medications by cloning mutated genes.
The American Medical Association holds 4 points of reasoning why cloning should not take place. They are: 1. There are unknown physical harms introduced by cloning. 2. Unknown psychological harms introduced by cloning, including violations of autonomy and privacy. 3. Impacts on familial and societal relations. And 4. Potential effects on the human genome. As we have not extensively tested cloning and it is a new discovery, there may be many side effects for the clone and for generations to come. The side effects could be physical or mental, because the basic foundation, DNA, could be changed somehow in the cloning process. Cloning could also bring about negative changes in society. People around clones or affected by them may feel that their rights have been violated. Clones may be treated as second-class citizens if they are treated as citizens at all. Certain questions regarding clones and the law may arise. Can a clone be considered the next of kin concerning wills or power share? Will clones serve in the military? Clones will probably be discriminated against by those who do not understand them or by those who fear them. Clones could also have an effect on the entire human genome in the future. Cloning could bring about changes in genes in future generations, eventually changing great medical uses. The concept can make it easy to get organ transplants, saving lives. In my personal opinion, though, the unexplored possibilities of negative side effects outweigh the medical opportunities posed to bring about the ugliness in our society and the unanswered reflections about where clones fit in the world.
The innovation and debate over cloning is something that has evolved and will continue to develop for centuries to come. What we do and what happens to that technology will be something we and future generations will have to deal with, and the choice is in our hands.